Here's if you're struggling with question 4 in the bioinformatics tool. Well, I guess 4 and 5. Well, let's start with 4. Because that one is kind of short. All right. So in question 4, I'm saying, imagine that that first ATG after the Tata box, and requires the Tata box, are marked here in yellow to draw attention to the Tata box. And if you're like, ooh, what is the Tata box again? Okay, that's a DNA sequence to which the RNA polymerase and the transcription factors will attach. So they glue on this sequence or around this sequence, and then they will scan this, and there will be transcription that will start here, but the translation will actually starts here, right? So I'm saying, let's assume that this ATG is the start codon for that gene. And so I'm asking you, which amino acid will be the 6th residue? So to answer this, I want you to know that residue is another word for amino acid when that amino acid is in a sequence, in a protein sequence or in a peptide sequence. So which one is the 6th? All I wanted is for you to figure out how to look at this sequence, and I wanted you to know that residue is a word for amino acid. So the methionine is your first amino acid. So it's the ATG, and it's not only the start codon, but it actually encodes an amino acid. The stop codon doesn't encode the last amino acid. It's just like no amino acid there. Okay, so first, second, and third, fourth, fifth, and 6 is the GAT. So you either put that on XPASI or you look up a genetic code, and then you can see what GAT encodes. But with this exercise, what I wanted for you to figure out is that ATG is the start codon and is the first amino acid in a protein, and that the word residue means amino acid in a DNA sequence, in a protein sequence.